Today, I have the privilege and the pleasure to present on behalf of my co-investigator the results of genome editing targeting BCL11A in patients with either thalassemia or sickle cell disease. And these are my disclosures. Few introductory background remarks just to explain the rationale at the basis of this innovative therapeutic approach. When a child with either thalassemia or sickle cell disease is born, it doesn't initially need a transfusion or develop the symptoms related to the disease. And the reason for this is that the patient benefits from the residual synthesis of hemoglobin F, which is the predominant hemoglobin during the fetal life. And there is also an inherited condition called hereditary persistence of hemoglobin F, in which patients, despite the fact that they can inherit also the thalassemia gene or the sickle cell disease gene, do not develop the clinical signs of birth condition because of this increase hemoglobin F. And we know very well that a molecule coded by a gene located on chromosome 2, namely BCL11A, is responsible for the silencing and the synthesis of the gamma chain. So the line of reasoning at the basis of the approach was that of inactivating this gene through the CRISPR-Cas9 genome editing approach with a specific erythroid-restricted inactivation of the production of BCL11A. After two seminal cases published at the end of 2021 in the New England Journal of Medicine, further patients were recruited into two trials, namely the CLIN-THAL 111 or the CLIN sickle cell disease 121, which are phase 1, 2, and 3 international multi-center open-label single-arm pivotal study of this drug product, formerly known as ctx001 and now called exa-cel. The primary endpoint for both studies reported in this cartoon, and you can see that the goal was that of estimating the proportion of patients who become transfusion independent if affected by thalassemia or free from severe vaso-occlusive disease if affected by sickle cell disease. And the age range of patients recruited is between 12 and 35 years, and both studies were planned to recruit up to 45 patients, and the enrollment is already completed. Today, I'd like to present the data on all patients who were infused as of February 2022, namely 44 patients with transfusion-dependent thalassemia and 31 affected by sickle cell disease. In terms of efficacy, 42 out of the 44 patients, namely 95%, reached transfusion independence. And the two patients who didn't reach transfusion independence had a 75 and 89% reduction in the transfusion volume. And the results are sustained over time. You see that, for example, in this first patient, the follow-up is even longer than 3 years. In the 31 patients affected by sickle cell disease, the success rate was 100% because all the patients were vaso-occlusive-free after exa-cel infusion with a duration of the follow-up from 2 to 32.3 months. In terms of safety profile, it is consistent with that of a busulfan-based myeloablation followed by the infusion of autologous stem cells. In particular, no patients died or developed secondary malignancies or myelodysplastic syndrome. There were 2 patients who developed an SAE, in particular, 1 patient with TDT had 3 severe adverse events related to exa-cel, many occurring in the context of secondary HLH, hemophagocytic lymphohistiocytosis, or macrophage activating syndrome. And the second patient had a delayed recovery of both neutrophil and platelets. So the safety profile of the approach is certainly reassuring for the patient. So in conclusion, the data from the 75 patients with either transfusion-dependent thalassemia or sickle cell disease showed that a single dose of exa-cel leads to early increase in hemoglobin F and total hemoglobin that are sustained over time. 95% of patients with TDT stop erythrocyte transfusion, and all the patients with sickle cell disease are free from vaso-occlusive crisis. The safety profile is consistent with that of an autologous transplant preceded by a myeloablation. And thus, we conclude that exa-cel has the potential to be the first CRISPR-Cas9-based therapy to provide a functional cure for patients with thalassemia or sickle cell disease. Thank you again for the opportunity to present the data.